points. Yeah, I will definitely come. That's great. Yes, thank you.
I hope
Nice.
you can.
Yes, of course.
Okay. So,
I think I have to start. Yes.
It's time to start. Good morning to everybody. We are going to start this plenary session on neuromuscular junction. There is a novelty. The Professor Ono wasn't able to attend personally, but the conference we will be presented by a video presentation. So we shall start. The first speaker is Professor Steve Barden from Skirball Institute NIY Medical School, and he will talk about the physiology and structure of neuromuscular junction. Please.
Good morning. So I'm going to spend the day trying to do two things. One, I'm going to spend a few minutes at the beginning giving you a brief introduction about the molecular mechanisms involved in the formation of nerve muscle synapses. This involves multiple steps. Initially, the recognition between the nerve and the muscle of one another, then an exchange of signals that allows the neuromuscular synapse to form during embryonic life. Subsequently, the steps that allow the synapse to mature and go through structural and functional changes, and then ultimately for the synapse to become stable and to be maintained throughout life. And I'm going to tell you about the main players that are involved in these pathways that regulate each of these three steps. And then I'm going to turn to more recent studies where we've tried to focus our attention on molecules that are involved selectively in the latter two steps, in the maturation and stabilization of neuromuscular synapses. So that's my goal today. So the synapse is, as you know, there's a single synapse on each muscle fiber, and at that site, it's very highly specialized, and it needs to be highly specialized to ensure for robust, rapid, and reliable synaptic transmission. And a key to that is the neurotransmitter receptors packaged in vesicles that are precisely aligned at active zones where they're in the readily releasable pool. And directly across from these active zones are very high concentrations of the neurotransmitter receptor, acetylcholine receptors. And the key molecules that are involved in specializing both the nerve terminal and the muscle fiber are illustrated here in this slide. So the key signaling protein that's derived from the nerve, from motor neurons, is a protein called agrin. It's released by motor nerve endings, and it's stably maintained in the synaptic basal lamina. It then binds to its muscle receptor, a transmembrane protein called LRP4. And the binding between agrin and LRP4 stimulates the association between LRP4 and a receptor tyrosine kinase, a muscle-specific tyrosine kinase, or a muscle synaptic tyrosine kinase called MuSK. So agrin binds LRP4, stimulates the association with MuSK, and simultaneously activates the kinase. Once the kinase is activated and phosphorylated, it recruits an adapter protein, DOC7, which is itself a dimer. So when it binds to MuSK, it facilitates the dimerization of MuSK. And DOC has two important roles. One is to stabilize the phosphorylation, the activation of MuSK, by keeping MuSK as a dimer, and also by signaling downstream from MuSK. The mechanisms by which the signaling is mediated downstream from MuSK and DOC are still very poorly understood. They lead to two pathways. One is a transcriptional pathway that results in an upregulation of genes that encode for proteins that are concentrated at synapses. So in fact, all of the proteins here, the genes that encode these proteins, are expressed at very high levels in nuclei that are near the synapse and at much lower levels in other nuclei throughout the muscle fiber. In addition to this pathway, there's a post-translational pathway that results in the redistribution and anchoring of key synaptic proteins, including LRP4, MuSK, and the acetylcholine receptor. And key to that clustering, so all the proteins shown here, with the exception of this protein, rhabsin, function in both pathways, the transcriptional pathway and the post-translational anchoring pathway. Rhabsin sits alone in that it's involved only in the anchoring pathway, not in the transcriptional pathway. And it acts to anchor acetylcholine receptors and a variety of other proteins at the synaptic site. So what I'd like to do today is we became very interested, as I mentioned at the beginning, in the pathways that are involved in the maturation and stabilization of the synapse. All of the proteins shown here are involved in all three steps, that initial recognition of nerve and muscle and the formation of synapses, as well as these later maturation and stabilization steps. And in the absence of any one of these genes, synapses fail to form in mouse models, and that results in embryonic lethality from respiratory failure. And in mutations in these genes that impair their function, that don't abolish them, that are hypomorphic mutations, result in disease. And this you'll hear about from Dr. Ono, because these are called congenital myasthenic syndromes. And autoantibodies against certain proteins in this complex, notably the acetylcholine receptor and MuSK, are responsible for the autoimmune disease myasthenia gravis. And you'll hear about this from Angela Vincent. So we've been interested, as I said, in the pathways that involved in strictly the later steps in synapse formation, in maturation and stabilization. And that's kind of illustrated in this cartoon here. So in the very initial steps in formation, the nerve contacts the muscle, and you form a very simple ovoid-like plaque. That's a very simplified ending that functions, but it's poorly developed. And in the mouse, over the next month, this synapse matures and becomes arborized to form a complex structure. There are a variety of other, in addition to these structural changes, functional changes that occur in the synapse. So this is so-called plaque to pretzel transition. The acetylcholine receptor turnover rate changes from a very rapid rate to a slow rate. The post-junctional folds, these invaginations of the postsynaptic membrane that allow more receptors to be, acetylcholine receptors to be packed into it, develop. And the muscle fiber, the synapses go through a period of synapse elimination where instead of being multiply innervated, they become singly innervated. And then this situation continues throughout the duration of life, in the case of a mouse, for over 2 years. So we're interested in what controls these latter two steps. And as I mentioned, all of the genes that I described in the earlier slide are involved in every single step here. But we had reason to believe that there might be genes that are involved selectively in the latter two steps, in these maturation steps, stabilization steps, but not necessarily in the formation step. And we decided, in a way to identify these gene products, we decided to focus our attention on this molecule, rhabsin, because we believed it would serve as a scaffold not only for the formation of synapses, but perhaps as a scaffold for proteins that are involved in these latter maturation and stabilization steps. So to remind you, mice that lack rhabsin, like the other genes, LRP4 and MuSK, they fail to form synapses. In a rhabsin mutant mouse, instead of acetylcholine receptors being clustered in the middle of the muscle, the acetylcholine receptors are expressed, but they're expressed uniformly over the length of the muscle. Rhabsin is about a 43 kilodalton protein. About the N-terminal half of it is made of tetratricopeptide repeats, 7 of them. It's followed by a coil-coil domain that binds the acetylcholine receptor, and it has a ring-like finger that binds beta-dystroglycan. So when synapses form, even before they form, even before agrin is applied and MuSK is activated, rhabsin is bound to the acetylcholine receptor. Once agrin stimulates MuSK, acetylcholine receptors become clustered, and they require rhabsin to do so. Once the acetylcholine receptor becomes tyrosine phosphorylated, the number of rhabsin molecules that are bound to the acetylcholine receptor increase, and this leads to the stabilization of otherwise labile acetylcholine receptor clusters. So there's a stabilization step that's associated with increased phosphorylation of the acetylcholine receptor and increased recruitment of rhabsin. So our approach to try to identify proteins that associate with rhabsin and might be involved in these maturation and stabilization steps are shown in illustrated in the next 2 slides. So our approach was to isolate acetylcholine receptors from the mouse, from early postnatal stages, as well as from mouse muscle cell lines. And we used biotin conjugated alpha-bungler toxin to purify the acetylcholine receptor with rapsin attached and with the presumed rapsin-associated proteins. And to identify proteins that depend upon rapsin for their association, we compared the proteins that associated, as analyzed by mass spectrometry, with acetylcholine receptors isolated from rapsin mutant muscle, again, from mice or from muscle cell lines that lack rapsin. So we're comparing the two. And we did many controls to ensure that the proteins that we were identifying were that co-isolated with acetylcholine receptor in a rapsin-dependent manner. And some of them are illustrated here. So we considered as background proteins that were attached to the beads in the absence of bungler toxin, or proteins that isolated with the beads in which we've blocked the bungler toxin binding sites with cold, non-biotin labeled bungler toxin before adding the beads. So anything bound to these was considered non-specific. Again, we compared the receptors with rapsin and without rapsin, and we also took rapsin mutant muscle cells and restored rapsin expression, and we required that these proteins that were attached here and not here were restored when we put rapsin back in. So all these criteria needed to be fulfilled in order for us to consider these proteins as an acetylcholine receptor associated in a rapsin-dependent manner. And I'm going to spend most of my time talking to you about one such protein. I'm also going to mention a protein that binds to acetylcholine receptor complexes, but it does so independent of rapsin. It binds directly to the acetylcholine receptor. Okay, so these are the proteins that we identified as binding to acetylcholine receptors in a rapsin-dependent manner. And what I'm expressing here is the amount of these proteins that were attached to receptor in wild-type cells over that amount attached or found in rapsin mutant muscle cells. And the acetylcholine receptor, of course, is present independent of rapsin. We isolate acetylcholine receptor, so that's set as one. And we find 3 proteins that have been identified in earlier studies as rapsin interactors, and those are alpha-actinin, beta-catenin, and calpain. And you can see these shown here in red. And they range to about a 5-10-fold increase in their association when rapsin's present. And then we found 5 new proteins. One is an old friend, and a surprising old friend to find in this context, and that's topoisomerase 2-beta that we studied over 15 years ago. And then of the other 4 proteins, there's one that we've studied in some detail, and that's what I'm going to describe to you today, and that's called MASC1. So that's microtubule actin factor 1. So you'll see this is a protein that binds microtubules, has multiple spectrin-like repeats, and also binds actin. So as you can see, there's about a 10-fold increase in the association of this protein with acetylcholine receptors that depends upon rapsin. So we validated that this protein, MACF1, is a acetylcholine receptor complex protein by showing that by Western blotting, if we isolate acetylcholine receptors from wild-type muscle, you can see MACF1 together with rapsin. And if we isolate acetylcholine receptors from rapsin mutant cells, of course, acetylcholine receptors are there. Of course, rapsin's gone, and so is MACF1. You can see this by immunostaining as well, that it's present at wild-type synapses. So this is adult muscle in which we've stained for MACF1. As you can see, it co-distributes with acetylcholine receptor, and if we denervate the muscle, cut cross sections, you can see that the staining persists even though the nerve terminal is gone. So it's a postsynaptic protein at the nerve-muscle junction. We're able to look at more detail about its location and demonstrate that it is exclusively postsynaptic and not presynaptic by optically sectioning through the neuromuscular junction. So we took images, actually not unlike these here, and then we optically section them, and this is a cartoon of that, so that we can see the proteins in the nerve terminal and those in the muscle by optical sectioning, and here's the controls for that. So synapsin, a presynaptic protein, acetylcholine receptor, you can see they're well separated. Bassoon, a protein in the active zone, acetylcholine receptor, easily well separated, pre and postsynaptic compartments. And you can see MACF1, it intermingles with the acetylcholine receptor. It's postsynaptic, we don't see any evidence of it presynaptic. I told you that there was one protein that we found that co-isolated with the acetylcholine receptors, but it did so in a rhabsin-independent manner. And I'm going to spend a minute or two here and a bit later telling you about the phenotype of this mutant in vizatin later. So as you can see here, if we isolate acetylcholine receptors from a wild type, again, we find the acetylcholine receptor. Alpha-actinin co-isolates in a rhabsin-dependent manner. And here's this protein vizatin. And it co-isolates with acetylcholine receptors from wild type and from rhabsin mutants. In fact, there's more of it, about twofold increase in the amount of vizatin that associates with ACH receptors in the absence of vizatin. And I'll give you some of the evidence and just tell you this is because it binds to the acetylcholine receptor in the same region that binds rhabsin. So rhabsin binds to multiple subunits in the acetylcholine receptor, predominantly the beta subunit, and so does vizatin. It binds directly to the beta subunit, and it binds to the same region on the beta subunit that rhabsin does. In addition, vizatin binds to the delta subunit, so it can bind simultaneously to the acetylcholine receptor with rhabsin, and it, as you can see in this here, and we've shown in other biochemical studies, competes for binding with rhabsin to the beta subunit. So it binds to the acetylcholine receptor independent of rhabsin, and you can see that it, like MACF1, is highly concentrated at the nerve synapse, single muscle fiber stained with vizatin, acetylcholine receptor. They're denervated, so the nerve terminal is gone. So it's indeed a postsynaptic protein. And here's just a small illustration of what vizatin looks like. It's a two-pass transmembrane protein. It's about 750 kilodaltons. 750 amino acids. Most of the protein is intracellular. There's only about 8 amino acids. It's extracellular. And this large C-terminal intracellular region is known to bind to proteins that contain firm domains, whether they're in rhodixin or in myosin 5A. So it binds any protein with a firm domain. And it also binds the protein ARF6, which regulates membrane trafficking and membrane curvature. Okay, so let me come back to MACF1. I told you it binds microtubules, and I told you it binds actin. So this is a cartoon of MACF1. It is an enormous protein. It's 650 kilodaltons. It has its actin binding regions at its N-terminus, its microtubule binding region, that is through EB1, the N-binding protein, at its C-terminus, and multiple spectrin-like repeats. So it binds EB1, as I said, therefore it binds microtubules, and presumably has the capability of binding any other microtubule-associated protein. It had been identified over 10 years ago in a 2-hybrid screen by Bob Bloch and his colleagues as a protein that interacts with rhabsin. So this wasn't the first evidence, but it had been ignored, and there had been no follow-up studies other than it bound rhabsin in the 2-hybrid screen. And Bob Bloch showed in the 2-hybrid screen that it was the TPR region of the rhabsin that binds MACF1, and it does so through the actin binding region. So it's expressed in a wide number of cells. It's widely expressed in the body, and straight knockouts of MACF1 result in embryonic lethality due to a failure to implant. So we needed to bypass this problem, so we made muscle conditional mutants in MACF1. Okay, so we made MACF1 mutants. They're viable. So unlike LRP4 MuSK and agrin and rhabsin that die at birth, these don't die at birth, and as you see, they'll form synapses. Nonetheless, we were able to show that we were able to reduce the expression of MACF1. This is an actin-Cre line that inactivates MACF1 from muscle after muscle fibers have formed. It leads to about a 10-fold reduction in MACF1 expression in muscle tissue and about a 95% reduction in skeletal muscle cells within skeletal muscle tissue, as shown here. And by staining, you can see MACF1 is present at synapses in the postsynaptic membrane and control muscle cells that control synapses, but in these mutants, it's gone. Synapses, as you might expect, since they're viable postnatally, form. And the synapses largely look the same. So they're the same number of synapses, the size of the synapses is the same. There's a 20% reduction in the density of acetylcholine receptors at individual synapses. But at birth, that's the only defect that we've been able to see so far. Now, I told you that synapses form, and we've been looking at this stage here right around birth, they're largely normal, with the exception of this small decrease in the density of acetylcholine receptors, but there are these important steps that occur later, both maturation and maintenance. So we decided to look later and ask whether these synapses continue to mature and whether they're maintained. So what you can see is that P180, several months after birth, synapses, and I'll give you the quantitative data for this, increase in size, they arborize, they go through these structural and functional changes that I mentioned earlier. And this does not occur normally in the MACF1 mutants. You see large numbers of synapses that are exceedingly small, large numbers of synapses where the synapse is fragmented into bits and pieces rather than one continuous arbor. So to give you a sense of quantitation, these are control wild type or control mice. After birth, the size of the synapse increases dramatically during the first month, and then it continues to increase more modestly over the next several months. The density of receptors is relatively constant. There's always co-localization of nerve terminals and acetylcholine receptors throughout time. And there's, as I said, very little of any fragmentation. The synapse is usually a continuous structure. In the MACF1 mutants, again, the size is normal early, and it continues to be normal to about a month, but then it stops growing, and the synapses become smaller with time. There are always nerve terminals perfectly co-localized with this smaller postsynaptic membrane. There's always perfect co-localization. The density is reduced early and stays lower throughout time. And then this continuous or fragmentation becomes worse and worse with time. So as again, in wild type, there's very little fragmentation. It's all continuous, and the number of fragments increases dramatically with time in the MACF1 muscle conditional mutants. We measured the behavior of these mice. And at P30, when the synapses are largely normal, there's little, if any, change in the old limb grip strength or their behavior on the rotorod. There's a bit of a change. So I'm showing you data for males and females here. And this is males here and females here. So females, there's a bit of a difference at P30, but it's pretty subtle. And in males, there is no difference at P30. But by P180, there's a difference in grip strength for males and females, and a significant difference in their behavior on the accelerating rotorod for males and females. So they start to show behavioral differences when the morphological changes of the synapse are most striking. The muscle fibers, as you might expect, if they become less mobile and less active, become atrophic. So again, at P30, the size of the muscle fibers is normal, but by P180, when they're showing defects in motor performance, there's about a 30% decrease in the cross-sectional area of the muscle fibers. And we believe this is due to defects in synaptic transmission. So if you record miniature end plate potentials from the muscle, you see about a 20% decrease in the frequency of the MEPs, which is proportional to the decrease in the synaptic size, as they should be correlated with one another. And there's also, as I mentioned, there's a decrease in density of ACH receptors. The muscle fibers being smaller have a higher input resistance, so there's a greater voltage for a constant current, but the quantal content is also reduced. So there's a decrease in quantal content and a decrease in mini frequency. And we think it's these changes in basal synaptic transmission, and we haven't stimulated at high frequencies, the kind that would normally be ongoing in vivo in the mouse. But we think these are the changes that underlie the behavioral changes and the atrophy of the muscle. But there's still much more to be done to understand how these physiological changes are occurring and how MACF1 regulates synaptic stabilization. And I'll come back to how we think it might work in a minute or two. But I wanted to turn briefly to vizatin, the protein that I said that binds directly to acetylcholine receptors. So we believe that MACF1 isn't required for the very early steps in formation. It isn't even required for this first steps in maturation, but it's required in some way in the stabilization step. Vizatin, so we did the same kind of experiments. In activate vizatin, it's required early in development for implantation, so we knocked it out selectively in muscle, and then we looked at synapses, and those failed to mature. So those are defective at this earlier, this intermediate step. So again, in wild type, you go from this transition from a plaque-like to a pretzel-like ending, and in the vizatin mutants, although some of them develop branch structures, many of them, more than half of them, remain in this immature state, poorly branched, highly fragmented. whereas the density of ACH receptors and the size remains relatively normal. But it's this shape change that is perturbed in the absence of vizatin. So let me come back to MACF1. So MACF1 is recruited to receptors in a rhabson-dependent manner, as is MAP1B. So MAP1B, you may recognize, is a microtubule-associated protein 1. It binds to microtubules. I've told you that MACF1 recruits microtubules, so you might imagine that the loss of MAP1B might be related to the loss of MACF1. So we wanted to test that idea. Does it bind directly to rhabson, or does it bind to MACF1? And that's why it's lost in the rhabson mutants. So we isolated acetylcholine receptors from wild type and from MACF1 conditional mutants. And as you can see, as you expect, MACF1 is lost, so is MAP1B. Rhabson is still there. ACH receptors are still there. All the proteins that bind directly to rhabson are still there, but MAP1B is gone. So we intuit, and you can see this also from microscopy, where it's not just MAP1B, but all of the microtubule-associated proteins. EB1, the M-binding protein that binds to the plus ends of microtubules, and beta-tubulin itself. So at wild-type synapses, again, you see MAP1B, EB1, and beta-tubulin both presynaptic and postsynaptic. And when you take away MACF1 from the muscle, the presynaptic stays, but the postsynaptic is gone. So all of these proteins that are part of the microtubule complex, EB1, MAP1B, microtubules itself, all depend upon MACF1 for their concentration at the synapse and their attachment indirectly to acetylcholine receptors. Okay, so this leads us to the idea that following the roles of aggrin and LRP4 must and dock in the formation of synapses and in their continual maturation and maintenance, you have proteins such as vizatin that attach directly to acetylcholine receptor, and as I told you, that's mediated through the beta and the delta subunit, that has a role in these maturation steps, these morphological changes that occur at the synapse postnatally. And then there are proteins MACF1 that are recruited to rhabson, and they then recruit both EB1, microtubules, MAP1B, actin, and they're involved in the stabilization of synapses. So we have at least a sense of some of the proteins that are involved in these very important steps. We still don't know how they work. And so there's much more to be done to understand how they work. And we wonder whether, of course, whether there are alterations in the function of these proteins that might underlie some of the diseases in humans that you think might sensitize, either themselves be involved in defects in synapse formation or contribute to alterations in the way the synapse works that may perturb human motor performance as well. So I think the big summary here is that the synapse maturation and stabilization do require molecules and mechanisms, including vizatin, MACF1, and its partners, that are distinct from those molecules and mechanisms that are used to form neuromuscular synapses. So there are some molecules that are used at every step, and some molecules that become recruited and activated and functional only during these later steps in synapse formation. And so I think this leads to a set of experiments for the future to try to identify other proteins that are involved in these maturation steps and to understand in more detail how they work. So there were two people who carried out these studies in my lab. All the work on MACF1 was done by a postdoctoral fellow, Julian Ori, and the work on vizatin that I briefly described to you, there's much more of that than I told you about, was done by a graduate student, Natasha Permal. So thank you very much for your attention. And I don't know what we're celebrating here.
Thank you very much, Professor Barden. You have been on time. So we have time for questions. Any questions?
So in all of your elegant diagrams of acetylcholine receptors, you show that even the clustered receptors have a gamma rather than an epsilon subunit. At what stage does the mature epsilon subunit substitute for the gamma subunit, and what pathways are related to that?
Yeah, so in the mouse, the transition occurs right around birth. So there's still some gamma, beginnings of epsilon at birth. With regard to vizatin, it binds, we didn't actually test binding to gamma, we tested binding to epsilon. So vizatin, as I said, binds beta and delta, and it does not bind alpha or epsilon. But the transition occurs during the maturation phase. That's when the post-junctional folds for epsilon is activated. Yeah.
Thank you very much. It's really nice talk. I'm always interested in complexes, and there are a number of questions which you didn't really address, and that's fair enough. So what are the proportions of those various proteins? Numerically, I thought that MAC-F1 was probably a bit lower than the others, but that may just be the blots. What are the interactions between them, and is phosphorylation involved in those interactions?
I am sorry, Angela, I'm not going to be able to really answer any of your questions. So the stoichiometry, we don't have, that's hard. You know, we can't use antibodies for that because of all the variabilities with antibodies. The mass spec has enough variation in its sample to sample from recovery that we don't have good quantitation there. We do, the only quantitation, we actually have some for vizatin because there we did SILAC experiments. And the ratio of vizatin to rhabsin is pretty much 1-to-1. And as you know, for rhabsin, as I mentioned early on, there's an increase in the amount of rhabsin that associates with ACH receptors following agrin stimulation. And we have pretty hard numbers on that. So that's about 1.5-fold, and it's the same for vizatin. For MACF1, I don't have good quantitative data. And phosphorylation data, I also don't have any data. I don't know.
So what keeps the complex together? Are there any actual structural bonds, or is it just
Between vizatin and ACH receptor, as I said, it binds to the main intracellular loop in the beta and the delta subunit. And we see binding, and so this is non-covalent binding, this is just reassociation binding, to two areas of the intracellular loop. And those are the same two areas that bind rhabsin, both the so-called N-terminal half of the loop as well as the constitutive C-terminal part of the loop. The same is true for delta, so the same areas of the loop. MACF1, we haven't studied in detail the binding. I think there's probably more detail that existed from this old paper from Bob Block that it binds the TPR repeats. And from Bob, he thought it was a subset of the TPR repeats, not all of them. And as I said, it binds to the actin binding domain, and there are two actin binding domains at the N-terminal region of MACF1, and whether it's one, the other, or both that are required, we don't know.
Great talk, Steve. Thanks very much. In looking at disease, we're always intrigued by the different muscle groups that are affected by different mutations. And I wondered whether you'd looked at different muscle groups in your mice to actually see whether you had any effects on fast or slow channel fibers or different muscles.
David, I'm going to disappoint you too. So, as you know, we largely look at the diaphragm muscle. And nearly all of the studies here have been done in the diaphragm, although some of the sectioning, some of the light microscopy has been done in the leg muscles. And obviously, the behavior that we're reading out is leg muscle dependent. But we haven't done a systematic analysis of different muscles and looked at the expression of the different players. We don't know. Yeah.
Actually, great talk. Thanks, Steve. There was a recent report on microtubule-associated proteins CLASP2 that are also involved at the neuromuscular synapse. Does MACF1 have any relationship to CLASP proteins?
Yeah, Marcus, I know that through the neuregulin or B path and dystrobrevin. So I don't know. We've only used antibodies to alpha and beta tubulin and haven't looked at subclasses, so I can't say. But that's it. We certainly are aware of that, and I think that's an interesting possibility, pretty likely possibility, it seems to me, yeah.
Hello, I have a question here. Regarding this marker, did you study them in the denervation, reinnervation processes in adult animals? And which one of them would you recommend to study to evaluate the formation of new neuromuscular junction?
I'm sorry, I'm having a hard time hearing up here.
Okay. Did you study all these markers in adult animals to evaluate the reinnervation processes?
Have we looked at reinnervation?
Yeah.
Very good. No, we have not.
And, and which one of those markers would you think would be the best to study?
Angela, can you help me again?
Which of the proteins might be the most important
In reinnervation, I don't have a clue. I don't have a clue. Wild speculation, just to be totally wild. I mean, I think it is worth making the point that for the MACF1 mutant, that when we see defects postsynaptically, which is what I showed you, and then you could see from the quantitative analysis that there was consistent co-localization of nerve terminals and muscle. This is a common theme. This is not unique here. But when there are changes postsynaptically, and we've just knocked it out from muscle, not from nerve, and we have no reason to believe MACF1's in the motor axon, there are changes in the motor axon. So there's this, again, crosstalk between what's going on in the muscle, and it's read and recognized by the nerve. Whether during reinnervation, the changes in the postsynaptic structure would be reimposed on the nerve. Interesting idea. I have no data for that. Yeah.
Thank you.
So the next.
So this fragmentation that you describe, is it in any way similar to this fragmentation that's supposed to happen during aging? I mean, and maybe it plays a role?
Yeah, good point. So that reminds me. Okay, so the point you bring up is fragmentation is often seen during aging or deteriorating synapses, and that's exactly one of the reasons we looked. And there are, in the visatin mutants, there are other changes that we see that are commonly seen during aging. That is axonal sprouting, thinning of axons, bulbous-like endings presynaptically. So we haven't seen this for MACF1, but for visatin mutants, there are many changes that resemble those that are seen at deteriorating aging synapses. Well, functionally, the role of fragmentation, I think this is very unclear whether it imposes functional changes on the synapse. In the visatin mutants, I should also mention there are dramatic changes in the organization of the post-junctional folds. And so again, they may have consequences in human, they may have, in mice, and they may have more dramatic consequences in humans.
Okay, any more questions?
Here's another one you might not be able to answer, but I think you just mentioned the post-junctional folds, and it's very intriguing, I think, how they are formed and why they are formed, and if you've got any clues to that, it would be interesting.
So this is one where I thought a bit about and have a clue. The changes we see in the visatin mutants are very similar, and we do see those also in the MACF1 that are resemble those that we described earlier in mutants where acetylcholine receptors can't be phosphorylated, where there are fewer folds and many fewer folds that have openings or mouths in the synaptic cleft. So visatin, as I said, one of the proteins that recruits is ARF6, and ARF6 is involved both in membrane trafficking and in controlling membrane curvature. So since the folds are highly curved structures, that's an intriguing possibility and one we thought we would test. Yeah.
Okay. Okay, thank you very much. So we move.
So as you already heard, Professor Ono will not be able to be here in person, but we will have a video presentation from him, and this is also possible to ask questions after the talk. And Professor Ono, he spent many years at the Mayo Clinic with Professor Engel researching on congenital myopathies, and now he's a professor at the Nayoga University Medical School, and we look very much forward on his talk. So, good morning, Mr. Ono.
Thank you for kind introduction. So I can start my talk. Okay. So I would like to apologize for not directly attending the meeting because I was not aware of the detailed and the strict passport regulations of the Schengen area. So I would also like to thank Dr. Griswold for giving me an opportunity to have an online talk about congenital neurasthenia. Let me start my talk. So today I'll talk about 5 stories. Let me start with the first one. First one is the recent advances in congenital myasthenic syndromes. So a total of 31 genes have been reported in CNS. And the mutant genes are shown in red letters, and the mutation reports are increasing in 4 groups. The first group is made of ACHR classic molecules. I think these molecules are explained in detail and precisely by Steve Varden, so I'll not touch on it. And the second group is made of glycosylation enzymes. And GFPT1 is a rate-limiting enzyme to make UDP. And GMPPB is an enzyme to make GDP mannose. These 2 products are used for N and O glycosylations, but now we see only N glycosylation. And DPAGT1, LG14, LG2, these are enzymes to make N glycosylation. But we don't know why mutations in these glycosylation enzymes affect neuromuscular junction. All of these are expressed ubiquitously in our body. And the third group is synaptic vesicle associated proteins. Choline is taken up by high affinity choline transporter, and it is converted to acetylcholine by choline acetyltransferase. And acetylcholine is taken up by vesicular acetylcholine transporter into synaptic vesicles. Rabifilin 3A involved in the trafficking and the release of synaptic vesicles. Okay. The last group is made of snare complex associated proteins. Synaptotamin is a calcium sensor, and the synaptobrevin, SNAP25, and syntaxin make a triple helix to fuse the synaptic vesicle to presynaptic membrane. MUNC31 is an accessory protein for syntaxin. So I'll talk about these 4 groups individually. Okay. The first one is acetylcholine crossing signals. Okay, so the mutations in these molecules show the limb girdle type muscle weakness, and in admin CMS, LEMS like Lambert-Eaton-like waxing of schema is observed. And interestingly, choline esterase inhibitors and 3,4-dienopyridines are not effective, or even worsens myasthenia. But the marked effect is observed in ephedrine and salbutamol. And Judith Rudolph at Heidelberg recently reported that muscle sympathetic nerve innervates the neuromuscular junction and enhances ACHR clustering. They show that the sympathectomy reduces the size of ACHR clusters and the size of CMAP, and the simultaneous administration of sympathomimetics rescues the phenotype. Okay, so I'll show you an example of a marked effect of ephedrine in a patient with AGRI mutation. Oops. So this is before ephedrine, and in about 4 weeks, the patient became like this. And so this is before ephedrine. And he can move his hand against its gravity, and after treatment, it's almost normal. And he has difficulty in standing up from the chair, and after treatment, and he's like this. Let's look at how he walks. This is before treatment. And this is after treatment. So ephedrine has marked effect. Okay. So I'll show you what we reported in LRP4 mutations. The mutations in LRP4 have been reported in these bone diseases, CLSS and SOST2, and we found missense mutations in the third beta-propellar domain of LRP4, and interestingly, the other missense mutations are reported in LRP2. So we found that our mutations, CMS mutations, are at the periphery of the predicted the third beta-propellar domain, and the SOST mutations at the center of the domain. And we found that the CMS mutations shown in purple affects binding of LRP4 to agrin and also to MASC, and these mutations affect acetylcholine clustering. But the yellow mutations, the SOST2 mutations, did not affect the binding to agrin, MASC, or HHL clustering, but these mutations affected Wnt signaling in osteogenic cells. And we also made 4 artificial missense mutants, and they behaved as expected. So I move on to the second group, and second group is comprised of the glycosylation enzymes. And again, they show limb-girdle type weakness, and tubular aggregates are observed in these groups, but not in GMPPB CMS. And serum CK level is elevated in GMPPB, GFPT1, and also in slow channel syndrome. Slow channel syndrome is not in this group, but CK is also elevated in slow channel. And indeed, GMPPB is mutated in muscular dystrophy, dystroglycopathy type 14. Similarly, DPAM-T1 and ALD2 are mutated in congenital disorders of glycosylation types IJ and II. And the third group is made of synaptic vesicle associated proteins. The shared features are episodic apnea and impairment. The high-frequency nerve stimulation or exercise load are required to elicit the synaptic decrement. And the rapamycin-3 mutant, the increment is observed at high frequency, similar to LEMS. And putting SOS inhibitors are effective in these patients. And the last group is SNARE complex associated proteins. So electrophysiologically, these patients are similar to Lambert-Eaton-Myers syndrome, except for the SNAP25 CMS. And synaptotagmin mutant and SNAP25 mutants are autosomal dominant, like slow channel CMS. So I move on to the next talk. Next topic is about splicing. I'll show you 5 examples today. 2 of these are from the patient mutations, and 3 of these are normal splicing of neuromuscular junction molecules. Okay, I'll explain what kind of methods we use to dissect these splicing mechanisms. We start from the mutation in the patient or block scanning mutagenesis to identify the cis-acting element. Next, we isolate RNA binding protein by RNA affinity purification like this, and the isolated one is analyzed by mass spectrometry and confirmed the protein by Western blotting. We next perform the Western knockdown of RNA binding protein to see the effect on splicing. Then we introduce mutations at the cis-acting element one by one, and we also deplete the RNA binding protein by antibody to see the effect on the splice cell chromosome assembly. Then we also tease the RNA binding protein with MS2 hairpin or PP7 hairpin to prove that the RNA binding protein acts on the exact target. We also perform in vitro splicing analysis, and we also isolate splice cell complex to see the effect of the mutation or the effect of the deletion in this region. Finally, we perform in silico analysis to prove that what we found in a single gene that we found in CMS can be applied to multiple genes. So let me start with the first example. The CHRNA encodes acetylcholine alpha subunit. This gene has 75 nucleotide extra exon, and the inclusion of this exon generates non-functional subunit. And skipping of this exon generates functional subunit. In a muscle, in a human muscle, these two transcripts are generated in a ratio of 1:1. And we found that HNRPL binds to exon P3A, and this brings PTBP1 to suppress the splicing of this exon. And we also found that HNRPH binds to the intron, and this also induces exon skipping. In a patient with CMS, we found G2A mutation at intron position -8, and this mutation affected the binding of H, so this exon was included. In another patient, we found a G2A mutation in exon P3A, and with this mutation, L cannot bind to the target, and instead HNRPLL binds to the target, and LL cannot bind to PTBP1, and the splicing cannot be suppressed. Okay, the second example is collagen Q. We found an A2G mutation in a patient with congenital myasthenia, and in exon 16 of collagen Q, SRSF1 binds to the target, and this brings in one snap to splice this in, but this mutation displaces binding of SRSF1, and it brings HNRPH, and H suppresses splicing. So the patient has no exon 16. Okay, the following 3 examples are not patient mutations, these are normal splicing. And this is mask gene, and in human mask, we have Wnt binding frizzled CRD domain, and this is made of exons 10 and 11. This exon 10 is alternative spliced in human, and we found that HNBC brings L and YB1, and these 3 molecules cooperate together to induce skipping of this exon. So we humans generate non-functional mask subbing, but we don't know why we need non-function subbing. The fourth one is complicated. We worked on the acetylcholinesterase. The unsplicing factor, HNPH, and polyadenylation molecules, CSTF64, compete each other to bind to the same target. And if HNPH wins, then this activates the distal 3' splice site, and this lesion is skipped to generate the T isoform. This is expressed in muscle and the brain. And if CSTF64 wins, then the proximal poly-A site is activated, and we have H isoform, which is expressed in hematopoietic cells, and R isoform. The last example is DOC7, and in exon 4 of DOC7, there are two 5' splice sites. When the internal proximal site is activated, we have normal DOC7, and which is shown in blue, and we have normal DOC7. But if the internal distal site is activated, we have a truncated DOC7, and of course we have non-functional DOC7. And we found that SRSF1 displaces binding of U1 SNAP from the internal distal site, and U1 SNAP binds to the internal proximal site to generate the normal DOC7. Okay, I move on to the prediction of splicing mutations. So in a patient with congenital myasthenia, we found a T2A mutation in intron 5 of lapsin gene, and we found that this mutation causes skipping of exon 5 and exon 6. And by isolating splice cells, so we found that this mutation weakens the binding of U2F65 to the intron. And we found there is no tool to predict the splicing effect of intronic mutations. So we decided to make our own tool by ourselves. So it's a complicated one. So we extracted 105 features to dictate splicing strengths. And these included the information about the branch point sequence and the cognitive interact and the motifs of unbinding proteins. So we use the support vector machine modeling to generate a model, and the validation shows that sensitivity and specificity were like this. These are not to our satisfaction, but better than nothing. So we made the IntoSplice web server available online. So you type in the chromosome number and the genomic coordinate and the reference sequence and mutant sequence, and the server returns if the splicing is affected or not. And I'm showing, we now see 4 examples. So we validated our tool, so we introduced a series of A mutation in intron 5, and we found, so this is a patient mutation. We found that 3 of them cause exon skipping, and our tool predicted that 4 should be abnormally spliced. So we missed 1 mutation, but the sensitivity and specificity are like this. I'll not talk about the splice cell assay. Next, I'll talk about the prediction of pathogenicity of missense SNVs. In a patient with congenital myasthenia, we found a M2T mutation in the beta subunit, we expressed this mutant in hex cells, and we found that the expression level was normal. And the single channel recordings show that this mutation shortened the channel opening burst to 60% of normal. But when we started writing a paper, we unfortunately found that this mutation, or this SNP, is a common SNP in Japanese population, but not in other countries. So we didn't have this information when we started functional analysis, but when we started writing the paper, we found this information. So this is not a mutation, this is a common polymorphism in Japanese population. So we plotted the birth duration and the expression levels of 15 first channel mutants. And you can see that if the birth duration is less than 30%, it's pathogenic. But if the birth duration is less than 60%, the expression level must be less than 60% to be pathogenic. So our mutant, or our SNP, was out of these ranges. So this was a normal polymorphism. So this failure prompted us to generate our own model to predict the pathogenicity of missense mutants, genome-wide missense mutants. Our strategy is to make individual model for each mutation. For example, for M2T mutant that we observed in the beta subunit, we ordered the modeling system to differentiate 268 normal M2T SNP and 268 pathogenic M2T mutants. So we made a model and we validated a model by leave-one-out cross-validation methods. And this is the ROC curve. Our tool outperformed the other 26 other tools. Porphin2 and Shift are commonly used, but these tools frequently disappoint us, and you can appreciate the performances of these tools. But you can also see that recently developed tools, including ours, are much better than these tools. So we also validated our tool with 3 different data sets, and our tool was better than 26 other tools. And of course, we are latecomers, and we have a good way to outperform the others. And Porphin2, you can see that Porphin2 and Shift are like these. The vertical axis is cumulative Jordan's indices, and the Jordan's index is sensitivity plus 60 minus 1, so the maximum should be 3 for 3 data sets. So we made the web service program, but this is not yet open to the public because we have just submitted a paper. So you type in the chromosome number and genetic coordinate, and the server generates 3 mutations. And the predicted amino acid changes are shown here, and the server returns the predicted pathogenicity. So this is the beta subunit mutation, and our tool correctly predicted. So we didn't have to work on the expression studies. Okay. We also applied our tool to mutations and 17 normal SNPs in DOC7, and our tool correctly predicted pathogenicity in 20 mutations and correctly predicted that these SNPs are normal in 15 of them, but we missed 3 mutant and 2 normal SNPs. The sensitivity and specificity are like this, and these are similar to those involved in global analysis. Okay, now we are dependent on this tool to start the functional analysis of identified mutation. Okay, and let me talk about the new molecule induced clustering. So by laser capture microdispersion final code, we found that RSPO2 is highly expressed. So we confirmed it by in situ hybridization. We see that spinal motor neurons are stained by RSPO2. So RSPO2 knockout mouse dies perinatally because they cannot breathe. And we found that in the diaphragm, that acetylcholine receptor bandwidth is enlarged, and you can appreciate terminal arborization of phrenic nerve, and the acetylcholine receptor clusters are round, and the areas are increased. And you can see that the aglin can induce phosphorylation of mask, and that RSPO2 can also phosphorylate mask in the absence of aglin. And you can also appreciate that the RSPO2 alone can activate the mask phosphorylation pathway, and aglin activation activity of aglin like this, so it's less than aglin, but in this amount, the activity is about 80%. Okay. As the neuromuscular junction, synaptic cleft is widened and the number of synaptic vesicles is reduced. So RSPO2 is a secreted protein, so we try to identify the origin of RSPO2, so we express the RSPO2 specifically in muscle and specifically in spinal cord to determine the origin. And in RSPO2 knockout mouse, the acetylcholine receptor bands are widened and the areas are enlarged, and the number of secondary branches is reduced. And muscle-specific rescue of RSPO2 had no effect on this phenotype, but spinal motor neuron-specific rescue of RSPO2 actually this one overcorrected the phenotype, but it had no effect on the number of secondary branches. So RSPO2 is an important molecule to make the acetylcholine receptor clustering. So I didn't show you the data. The receptor for RSPO2 is LGR5, and we need LRP4 and mask to have the effect of RSPO2, but we don't need aglin. And in addition to aglin and RSPO2, Wnt is also important to make HHR clusters. So we worked on the downstream molecules of Wnt. I'll just show you a single slide. We found that FGF18 is a downstream molecule. It's upregulated by Wnt, and the Wnt deficient mouse die perinatally because, again, the mouse can't breathe, and this was surprising for us. When we looked at nerve terminal, we found a gigantic nerve terminal by abnormal aggregation of nerve terminal. So the size of nerve terminal is markedly increased, and the density of synaptic vesicle is markedly decreased, and the simplified postsynaptic force. So FGF18 is also an important molecule to make neuromuscular junction. So today I talked about 5 stories, and these are diversified stories, but all of them stemmed from CMS studies. So these works were made available by my colleagues, my current colleagues, and also by my ex-colleagues and my collaborators. And I would like to express my sincere thanks to Andy Engel for his support to our project. And thank you for your attention.
So Professor Ono, thank you very much for this fascinating talk. Thank you very much. So, and we have plenty of time for questions. So is there, yes,
I want to put a question. You have shown that it's possible to treat myasthenia with ephedrine. Do you think it's possible to treat somebody 10 or 20 years with such a drug?
Sorry, you can treat which patients?
You have shown that it is possible to treat myasthenia with ephedrine. Okay?
To treat the patients with ephedrine, and whether it's possible to do it for a longer time.
Yes, I think so. I showed you that the ephedrine is effective for patients in mutations in acetylcholine clustering signals. The effect of ephedrine for anti-myasthenia is reported in mouse and human. But I don't know if it's reported in anti-AChR myasthenia patients. And I didn't show you, but ephedrine is also effective for collagen Q deficient patients and the slow channel patients. And we don't know why, but the Rudolph's paper shows that sympathetic nerve innervates the neuromuscular junction and important for the modeling of the neuromuscular junction and in collagen Q deficient patients and the slow channel patients. They have abnormal neuromuscular junction structures, and ephedrine may help restructure the neuromuscular junction.
So is there another question? So may I ask you maybe for a little bit more practical approach to differentiate the presynaptic versus the postsynaptic syndromes, and how you do the workup of these patients?
Oh, it's a good question, but I don't have a good answer. It's always very difficult. I just picked up some typical features, and of course we have a lot of exceptions. And for example, the smear complex associated proteins, this is similar to LEMS, and labfilin 3A mutation should be included in both, because this one has both features. And synaptic vesicle associated proteins, mutations in these molecules show the episodic apnea and cognitive impairment, but not in all patients. And the episodic apnea and cognitive impairment are observed in other patients as well. So it's very difficult to tell the location of the mutation. But fortunately, the exome sequencing became cheaper and cheaper, and we can work on all genes, all 30 genes at the same time.
Thank you very much. Is there another question? So if not, thank you very much, and give a big applause to Professor Ono.
Thank you.
Okay. So we move to the next speaker. Well, it is my honor to present Professor Angela Wiesen, one of the scientific for the last few years, and in the autoimmunity of the neuromuscular transmission and other disorders. Thank you.
Thank you very much indeed, and it's a pleasure to be here. Very grateful to Wolfgang and the organizers for inviting me. I'm also quite a little bit nervous about talking after two really scientific talks, which were both excellent. And I'm not really going to give you very much science, but just to tell you some of the things that I think are still interesting about the neuromuscular junction. I do have a conflict of interest because I and the University of Oxford hold patents for some of the antibodies I'm going to be talking about. So what I want to talk about is myasthenia and the neuromuscular junction, MuSK antibody myasthenia in some more detail, acquired neuromyotonia, which is a sort of disease of the neuromuscular junction, and a new, relatively new syndrome called fetal acetylcholine receptor inactivation syndrome, which is rare as far as we know, but maybe more common than we think. So, of course, myasthenia gravis is now very well established and very well understood disease, or at least so most people think. It's over 40 years since the antibodies to the acetylcholine receptor were first discovered. This is a patient of John Newsom Davis's, who he studied in the 1970s, just when we were beginning to measure the acetylcholine receptor antibodies. A technique which John Lindstrom had established earlier. And of course, this disease is now treated effectively with cholinesterase inhibitors, with plasma exchange, thymectomy, and various immunotherapies, and most of the patients seem to do extremely well on those. At the bottom right, you can just see the neuromuscular junction, similar to what Steve Burden showed at the beginning, and of course, that is the site of where the antibodies act. So this is my rather Mickey Mouse cartoon of the neuromuscular junction, which I've always found very helpful. And just to remind you that acetylcholine is released, it binds when there's a nerve impulse allowing calcium to enter, the acetylcholine is released, it binds to the acetylcholine receptors, this opens the ion channel in the receptors, and you get a depolarization which you can measure as an end plate potential. And of course, the important thing is that that end plate potential has to be large enough to rise above the threshold for activation of the sodium channels, which, as you've already seen from Steve Burden's talk, are much lower down in the folds. And when those sodium channels open, then they self-propagate the action potential along the muscle fiber, and that leads to contraction. The fact that all of this happens in a millisecond or two is just quite unbelievable, but that's what's happening the whole time, just as I speak or any of you breathe or fidget. Now, we know also, of course, that a single spontaneous release of a vesicle causes a much smaller miniature end plate potential, but we don't really need to know about those for today's talk. In myasthenia gravis, the antibodies against the acetylcholine receptor, which you now see here from above, as it were, and there are 5 subunits around a central pore, and it's that pore which allows the ions to go through into the muscle, the ions. And there are 2 alpha subunits, and a large proportion of the antibodies bind to the alpha subunits, a proportion of those to the main immunogenic region, which is present on both alpha subunits. And so you see that the antibodies, which are typically divalent, can bind to 2 acetylcholine receptors at any one time. And this leads to the mechanisms by which those antibodies act. So there are 3 mechanisms. The most typical is for the antibodies to bind in the manner I've just shown you, to cross-link the acetylcholine receptors. And that leads to an internalization of those receptors. This is a normal antigenic modulation, it's called. And that leads to the destruction of those receptors. And so you get an overall loss of the acetylcholine receptor. But at the same time, those antibodies are IgG1 antibodies mostly. IgG1 is a very complement activating antibody, And so you get the immune complexes forming and the membrane attack complex forming, which causes damage to the membrane. And that's a very important mechanism because that damage is much less easy to repair than just replacing the acetylcholine receptors. Obviously, the antibodies could, and sometimes do, block the function of the acetylcholine receptor in a more specific way. And you'll see that that does happen sometimes, but it's probably not so common in most patients. So here's the cartoon again. Now you've got less folds, smaller folds, loss of acetylcholine receptors. Those are the main mechanisms, or at least we think so, but it may not be quite so simple. So these antibodies have caused internalization of some of the receptors and damage to the neuromuscular junction. And that means that the end-plate potential may not reach the threshold for activation of the remaining sodium channels. But one of the questions people used to ask, perhaps less frequently now, is why aren't all the patients the same? Why are different muscles affected? Why is there such variability from day to day? And I think there are a lot of reasons why that might be the case, but I want to just raise those in your heads. One of the things we know from previous work by Ruff and Lennon is that the loss of folds reduces the number of sodium channels, and of course that makes it more difficult to generate an action potential in the nerve fiber. So already the disease has actually made it worse than it would have been just by loss of receptors. But at the same time, muscles are clever, like most of our cells, and they try to replace the acetylcholine receptors, and that's been shown in a number of studies, of which 2 are mentioned here. So that's the muscle responding. But what is even more intriguing is that the nerve responds. And in myasthenia, typical myasthenia, with acetylcholine receptor antibodies, the motor nerve seems to compensate by releasing more acetylcholine, again, to try and override the defect of neuromuscular transmission. And what's intriguing about this is it implies that there is some sort of retrograde signaling occurring across the synapse, and we don't really understand that at all, although there are people who are very interested in this. So what determines whether there is transmission failure at a particular synapse in a particular muscle? There's the amount of acetylcholine receptor loss, the morphological damage with loss of sodium channels, compensatory changes. All of these may be different depending on the individual, their genetic makeup, etc. But quite apart from those, you have to think of the blood flow limiting the diffusion of antibodies and complement into the tiny, tiny space of the neuromuscular junction. And I think all of these factors make the weakness in myasthenia somewhat unpredictable. Okay, so a few things about myasthenia gravis with acetylcholine receptor antibodies. It's well known, of course, that many patients are young females. They often have a HLA-DR3 type phenotype, and males are not so common in the early years. But increasingly over the last 20, 30 years, people have recognized that there's a second group of myasthenia patients who are older. They're often males rather than females. And in fact, the ages go up and up and up. And now I know that there are patients of 100 years old who have been diagnosed with this disease for the first time. So myasthenia in the elderly is almost like a new disease. It has to be thought about when you see patients with weakness appearing for the first time in that age group. These are the patients who are typically thymectomized. And I just want to remind you that the thymectomy trial, which you're going to hear much more about later on today, was initiated by John Newsom-Davies, with whom I worked for 25 years, and who tragically died in an accident in 2007 after setting up the trial, which was continued by colleagues in America, Gil, Rick, who are both speaking today. And the trial, which you'll hear about from them, has been successful, which was very exciting and a great tribute to John's memory. because it showed that thymectomy improved the weakness in patients, the QMG scores, and reduced the requirement for prednisolone. So those are the two graphs. In both arms, you can see that the treated patients, thymectomized patients, did better, and they also required less steroids to keep them well. And this was very exciting, and I hope you'll all go to the talks later on today. So treatments for myasthenia are now becoming quite diverse. I think still people will respond pretty well to cholinesterase, steroids, azathioprine, but there are many new treatments. Those can affect the B cells directly by inhibiting some of the molecules which stimulate them. Of course, rituximab is used very widely for getting rid of the B cells, but really we want to try and attack this end of the immune system where the plasma cells are actually making the antibodies. And to do that, one needs much more antigen-specific inhibition or deletion of those cells, and it would be really great if one could achieve that in this and many other antibody-mediated diseases. Of course, FcRn inhibitors are reducing the levels of antibodies in patients, and they're proving to be quite useful. And of course, eculizumab, which is anti-complement, is also, and you've seen many posters here, being used now, and that's very effective at preventing the complement-mediated damage. So I want to turn now to the seronegative patients, the patients who don't have the acetylcholine receptor antibodies. Acetylcholine receptor antibodies are typically measured by radioimmunoprecipitation assay using receptors labeled with this lovely snake toxin. And those are the sort of results you get. But these patients don't have those antibodies, and so they must have something else. And there are now, as we all know, and you know from Steve Burden's talk, other molecules at the neuromuscular junction which are very important, and two of them are on the surface and would make very nice targets for antibodies. That's MuSK and LRP4. But before we talk about MuSK antibodies, just want to mention something because it's been quite important in the development of our ideas. We knew that antibodies can precipitate the acetylcholine receptor, but when they do that, they usually only hang on by one arm. And if the antibody wasn't very high affinity, that one arm might not be enough to pull it down, to pull down the receptor in the immunoprecipitation assay. On the other hand, if you express receptors in a cell like this, and you cluster them with rapsin, which you've heard about already today, then the receptors will be so close that the antibodies could bind by two arms at once. So even these relatively low affinity antibodies would be able to bind strongly to the clustered acetylcholine receptors, which could be what they do at the neuromuscular junction. And in fact, when we do this, so you can see the clustered receptors, this is the patient's antibodies binding now by two arms at once, and being detected with a red secondary antibody. And here are the cells. They're not green because I didn't put that image in, but they could be green because we put green fluorescent protein in them. And you can see on the surface of the cells these clusters of acetylcholine receptors where the antibodies are bound. And this was a patient who previously had been completely negative by the conventional radioprecipitation assay. So that's taught us that cell-based assays are useful, and we now use them for a wide range of different antigens in routine diagnostics. These clustered receptor antibodies are very similar to the typical ones. They're just not very strong at binding in solution. And they can be helpful in some patients, and particularly in differentiating in children between myasthenia and congenital myasthenic syndromes, because in children, myasthenia is not very common. When you see a child with weakness, you might well think it's a congenital myasthenic syndrome, but of course, first of all, exclude that it isn't myasthenia gravis. And these antibodies have been proven to be quite useful in that context. Now, here is a patient who has the MuSK antibodies. And this is a patient of John Newsom Davis's. And I probably won't go through the whole video because of time, but she's been spoken to and she's been asked to smile, and that's about the best she can do. You can see her face is rather flat in expression. She's got some ptosis of the eyelids. Her eye movements are not great, and her lips are flabby. She can't speak well because her tongue is very affected, and you'll see that in a moment. She was treated for many years with conventional myasthenia treatments, and somehow they never really broke through. She would have temporary improvement with plasma exchange, but never really got better. And I knew her when she was older, at this age, but she had been ill since she was actually 2.5. You can see now her palate doesn't move, her tongue is very furrowed because of atrophy. Actually, she was treated with rituximab late in her 20s, and she really has done quite well with that, although certainly not improved completely, but she feels better. Interesting things about myasthenia are it is different from acetylcholine receptor antibody myasthenia. For instance, when you do single fiber, EMG, if you use limb muscles, you tend to find negative results. You see that most of the results are normal, apart from in one patient, whereas in typical acetylcholine receptor antibody myasthenia, you would get results from the limb muscles. You have to use facial muscles to be certain of getting positive results with single fiber, and here they are. So clearly this is important, but why? We don't know. Is it because there's more MuSK expressed in the facial muscles or less? It might be a better target for the antibodies, or maybe that there isn't enough there in the first place. We don't know. Similarly, with the muscle atrophy, there is definitely a difference in muscle volumes in the facial muscles, as shown here for the tongue measurements in patients with the MuSK antibodies. You see their tongue volumes are less, and there's often high signal, which is probably fatty replacement of the atrophied muscle fibers. So it's an interesting disease, and there's something special about these facial and bulbar muscles in these patients. But this was a new association of MuSK antibodies. This is a child who's reported quite recently from, I can't read it now, and I know perfectly well who he is, but Bertold Schark in Germany. And she was a child who actually had presented 5 years previously, but hadn't been diagnosed as having MuSK myasthenia. She was age 12 by the time of diagnosis. You can see she's got a very bad scoliosis. Her muscles are myopathic, but she had MuSK antibody. And she's been treated with IVIG and steroids with a dramatic increase in strength. Intensive physiotherapy has partially resolved her scoliosis, as you can see on the right. And you can see her muscles look much better on the right. So that's a patient, a child who obviously nobody had thought of myasthenia in the first place, and yet she had it and apparently can still respond to treatments with considerable benefits. So that's something to be thinking about, particularly if you're a pediatrician, but perhaps also in other people. Another interesting thing about MuSK antibodies is the prevalence, the geographical prevalence, because it seems to be different in different populations. And these are data based on antibody testing for many different countries who sent their samples to me when there was no routine testing or no commercial test. And Niels-Erik Gilhus is here, and he'll remember when I went to Bergen and had to admit that none of their patients had MuSK antibodies, and this is because they're too far north, whereas as you go further south, particularly in the Mediterranean countries, MuSK antibodies are very frequent or relatively frequent. They probably go down subsequently, but that's not very clear yet because we don't have the right data. There's also a very strong HLA association, which comes from the Dutch group, and that's also been confirmed in other populations, so, or at least very similar HLA associations. So that's intriguing. It's a very different disease from myasthenia. But also, the antibodies are not IgG 1, 2, or 3. They're IgG 4 predominantly, although not entirely. So here's some data from our own lab, and you can see the IgG 4 levels in 3 different individuals. Those are the high bars, but collectively IgG1, 2, and 3 certainly could make some impact on the disease, but most of the emphasis has been on these IgG4 antibodies, and they're very intriguing because they are not divalent, they're monovalent for their antigen, and that's been shown in a number of studies, particularly this one from Inga Konechny a year or so ago. These antibodies to MuSK go around in hybrid form so that only one arm is specific for the antigen. And this means they can't cross-link antigens, they can't activate complement, they can't internalize MuSK. But we still need to think about these as well. So we know a lot of things about MuSK myasthenia, and I think I've mentioned most of them. Marked facial and bulbar atrophy involvement can be severe and need prompt treatment, often responds poorly to cholinesterase inhibitors. Rituximab seems to be effective. Frequency differs with latitude, and IgG4 mainly, which does not activate complement or cross-link AChRs. So what do MuSK antibodies actually do? Well, this is my very Mickey Mouse diagram of Steve Burden's and other people's work. So we know agrin is released from the motor nerve, that's the green things. They bind to LRP4. That causes clustering of LRP4 and MuSK phosphorylation. That recruits DOX7, and something happens, and the acetylcholine receptors cluster. That's very nice. What do the MuSK antibodies do? Well, we can measure MuSK antibodies in their effects in vitro with the C2-C12 mouse myotubes. So when you add agrin to these mouse myotubes, the acetylcholine receptors cluster in the manner that I've just diagrammatically shown you. And you can see these little clusters on the surface of those myotubes. If you put the myotubes in healthy IgG, this is what you see. If you put them in MuSK MG IgG antibodies, you see less clusters, and some of them are very sort of poorly formed. So the MuSK myasthenia antibodies clearly interfere with that process. And in fact, they do indeed block the interaction of LRP4 with MuSK. And these are specifically the IgG4 antibodies. So those seem to be, from this, the most effective and the most clear demonstration of pathogenicity. And that's work from the Dutch group as well as ourselves, with Steve Burden that collaborated with the Dutch. So here are the antibodies inhibiting the binding of LRP4 to MuSK. And that's what happens. You get dispersal of the acetylcholine receptors because there is no MuSK phosphorylation, no DOX7 interaction, and the receptors therefore disperse. And in fact, if you take MuSK IgG4 antibodies and inject them into mice, as Jan Rassuren's lab did, then you see a reduction in end plate potentials in the mice. And that was present when they injected IgG4, but not when they injected the IgG1 to 3 antibodies from the same patients. So this seemed to prove that IgG4 was the only active antibodies in these patients. But when we looked at those myotubes and we separated IgG4 and IgG3 antibodies from 2 patients, we found that the IgG1 to 3 antibodies also reduced the clusters. So somehow those antibodies that are not IgG4 are also having an effect in vitro. We don't know well yet whether they also act in vivo. So we have a situation where normally you have MuSK phosphorylated and you have DOX7 and you have clustering. But there is a phosphatase that dephosphorylates MuSK. And when it does that, the phosphorylation goes, DOX7 unbinds, and this is due to this phosphatase. So we thought that if we could inhibit the phosphatase, we might be able to restore MuSK phosphorylation and override the effect of the antibodies. And that's what's shown here in some data that is not yet published from Safe Huda and Michelangelo Cal. Michelangelo is here in the audience. And you can see that the phosphorylation of MuSK is increased substantially by this drug, which is a SHP2 inhibitor that we're using. So it's a phosphatase inhibitor. It increases MuSK phosphorylation and AChR clusters and reverses the effects of the antibodies. So if you just look down at the bottom, the red bars are the effects of the antibodies on clustering in C2, C12s, and the green bars are the same experiment, but done in the presence of the SHP2 inhibitor. And on the right, you can see the effects of the different subclasses of antibodies. So IgG1 to 3 inhibits clustering, and actually those are also restored to quite a large extent if you use the SHP2 inhibitor. And the IgG4 antibodies, as previously shown, inhibit clustering, and their effect is almost obliterated by the SHP2 inhibitor. So we hope that that might show that by modulating that pathway intracellularly, you can provide a rather more quick and effective treatment for patients with MuSK myasthenia. And that sort of approach might be useful in other neuromuscular junction disorders where there is a lack of acetylcholine receptors, and anything that can help bring them together at the right place at the neuromuscular junction might well be useful clinically. But what about other patients who don't have either MuSK antibody or acetylcholine receptor antibodies? Here is just an overview of the situation. There are now a lot of antibodies that have been measured and described in myasthenia gravis. The acetylcholine receptor antibodies include those that are measured conventionally and those that are measured with the clustered receptors cell-based assay that I've shown you. MuSK antibodies can also be measured with a MuSK cell-based assay, which we do in addition to the precipitation assay. And then LRP4 antibodies are present in some patients, and agrin antibodies have been reported in some patients, and we agree that they are present in a proportion. In our experience, these are usually rather low levels and quite often associated with either MuSK or AChR antibodies, and perhaps are a secondary effect of the immunization, or the self-immunization in many patients. LRP4 antibodies we find pretty rarely. Cortactin is another antibody, which is actually a downstream molecule within the muscle. So the antibodies are probably not pathogenic, but they seem to be present in quite a high proportion of patients, and maybe a marker of disease, which could be useful in those patients who don't have any of the other antibodies. And then there's titin antibodies, which are mostly thymoma and old onset myasthenia related, and the KV1.4 antibodies, which are more likely in thymoma and various patients with typical myasthenia. So you can see it's become a quite complicated topic, and that's why I've really chosen to speak only about MuSK and AChR, because once you get into the others, there's too much. Because I think I'll just miss that one. I wanted to go on to something about neuromyotonia, because this is a video which, again, is really quite an old one. It actually came out first in Matthew Kiernan's journal, which he keeps pushing. I don't know if Matthew's still here, but he keeps telling people about publishing in JNMP. So this is a paper that was accepted by JNMP before Matthew took over. This is spontaneous muscle activity due to peripheral nerve hyperexcitability. Some of the patients have anxiety, sleep disturbance, depression. It improves after plasma exchange and steroids, for instance. And we described, well, Ian Hart and ourselves described in the 1990s, and then subsequently in this paper, that about 40% of patients have what we thought was the potassium channel complex or potassium channel antibodies. We were wrong. The antibodies do not bind to the potassium channels themselves, at least certainly not on the outside, the bit where they would be pathogenic, but they bind to other proteins that are part of the potassium channel complex. And this is why I'm interested in complexes, because actually I think those complexes are going to be more and more important in everything that we do. So here are the potassium channels. They are modified and localized by the existence of these other proteins that control their localization in nerves and also their activity. And the fact that we were wrong here, I've always said was, I thought, rather a good thing, because we learnt much more by studying this. And then I saw very recently in an editorial type paper in Brain by Andrew Lees, this quote from Gowers, who was a very famous neurologist in Britain: "Gentlemen, it is always pleasant to be right, but it's generally a much more useful thing to be wrong." So don't worry about being wrong, it's always useful, as long as you try and find out why you were wrong. I think that's a nice quote. So we've been looking at patients with neuromyotonia. They were collected by Matthew Kiernan in Sydney and Osamu Watanabe and Dr. Arimura in Kagoshima. And what I wanted to draw your attention to was not the muscle symptoms, which you're probably all aware of, but the autonomic disturbance, which was present in 47%, pain or paresthesia, or both, in 53%, and other CNS features in 29%. So this is not just a peripheral nerve disease. And you have to realize that even with the neuromuscular junction, there are patients with central nervous system disorders. Most of the patients get better, a few only with anti-epileptic drugs, but usually with immunotherapies. So this shows modified ranking scores. Some of them are quite severe to begin with, but with treatments tend to get better, as you can see. So what antibodies do they actually have? Well, it's complicated, and not many of them have them. In fact, in our experience, it was 17 out of 38, slightly under 50%, about 40%. And the antibody levels are not necessarily very high, but they're positive in this number of patients. And the interesting patients are the ones that have both Caspr2 and LGI1 antibodies. And it's fairly unusual for patients to have both of those together. And usually it means they've either got Morvan syndrome, which is very rare, or neuromyotonia. It didn't seem to make much difference whether they had antibodies that we could detect or not. And that's important. So seronegativity in this disease shouldn't be taken too seriously. They may still have an immunotherapy responsive disorder, which is shown here. So these antibodies, patients who have both antibodies, Caspr2 and LGI1, there were only 6 of them. 5 were male. 5 of them had pain. 6 had autonomic disturbance. 5 had sleep disturbance. And 4 altogether had psychiatric disturbance. And in fact, 2 of them had tried or thought of committing suicide. So depression is really quite an important thing in this disease. And 3 of them had thymomas, 2 of them invasive, 1 with acetylcholine receptor antibodies, so myasthenia came in there. And in fact, prostate cancers seem to be more common than you might expect in a similar population. So the antibodies are not incredibly important in neuromyotonia. I think you can manage without them. Electrophysiology is a very good test for this, but they are intriguing in terms of what they mean to the patient. And one of the things they mean is pain. And this was a pain survey that we conducted totally independently of the physician patients. This was done by a patient through his own website. They did a survey of pain. Pain was most common in the lower limbs and feet, which is what had been reported, but was also present in other parts of the body, including the head and the neck. It was described as aching, burning, electric shocks, and cramps. It could be very severe at times, but most of the patients appreciated that they had responded to treatments. But nevertheless, it often greatly interfered with their quality of life, and that was all aspects of life. Now, this was a very unorthodox patient questionnaire, but I think it makes you realize, I hope, that if you see patients with these peripheral nerve disorders, you really do have to ask about pain, not just wait for them to tell you. And in fact, this is confirmed by the study that David Bennett talked about yesterday, which I hope some of you were at, where he showed that either immune or genetic mediated disruption of Caspar-2 causes pain hypersensitivity, and it's due to enhanced primary afferent excitability. And this just shows you some of the data, which I won't go through. He explained it very nicely, much better than I can. But this means that antibodies and pain are beginning to be something which I think should be recognized. And of course, you've already heard a lot about nodal proteins and pain. Caspar-2 is a juxtaparanodal protein. So it comes within the same sort of spectrum. Can I have another few minutes, couple of minutes? So I want to just go back to acetylcholine receptors to finish off with. In the past, some of you may have come across this situation where you have babies that are born with arthrogryposis, where the mother usually has myasthenia gravis, and the antibodies cross the placenta. Normally, the effect on the baby, if it's anything at all, is transient, and the baby is weak at birth, but then gets better over 2 or 3 weeks. Sometimes they need an exchange transfusion. But in the 1990s, David Hilton Jones and colleagues came across this case where a mother had had 4 consecutive babies with arthrogryposis that was so severe, this is multiple bent joints, so severe that in fact 3 of them died at birth or shortly afterwards, and only 1 survived, and that child had joint problems which were very difficult to handle. It was only after that history that somebody realized that the mother had myasthenia. She had high antibodies to the acetylcholine receptor. She was treated with thymectomy and immunotherapies, and then had a child who was only very slightly affected. So there was something in her antibodies that seemed to be causing this condition. And in fact, she has antibodies which bound not entirely, not only, but the exciting antibody was the one that bound only to the gamma subunit, which is present in the fetal form of the acetylcholine receptor, which I didn't show you before. This is the adult form with an epsilon. Here it's the gamma form, which is a fetal form. And these antibodies appear to bind specifically to the gamma subunit, but in such a way that they can block the activity of that acetylcholine binding site. So these antibodies block function of the fetal receptor, and when they cross the placenta, they paralyze the baby, and then it develops these joint contractures which can become permanent. So this is a very intriguing syndrome, but also some of the mothers turn out to be asymptomatic, presumably because they've got mostly fetal acetylcholine receptor antibodies and very few of the adult receptor antibodies. So you really have to think about this in babies born in that sense. We were very intrigued by this whole idea of maternal antibodies causing developmental disorders, and we set up an animal model where you inject the pregnant mice dams with patient's antibodies, and if they're from a mother who has had children affected with arthrogryposis multiplex congenita, then the babies are often born with fixed joints, they're partially paralyzed, they stop breathing, and they die after birth. Whereas this is a healthy baby born to a mother injected with control antibodies. So we had this model, and you'll see that we've just used it very recently, because some of those children don't die at birth, obviously, and sometimes they have minor arthrogryposis, but they don't recover full function. And this was a child who had what we're now calling maternal antibodies to the fetal acetylcholine receptor, and it's called this fetal acetylcholine receptor inactivation syndrome. And I didn't coin that name, but it has been coined. I'm not quite sure by who. So this child had maternal antibodies to fetal receptor, and they were associated with muscle weakness and hypotonia, but this persisted after birth. And you can see his face here. It looks like a congenital myasthenic child, in fact, I would have thought. But what was very intriguing is he improved with salbutamol. And as you'll hear later on, and you've already heard about ephedrine, adrenergic agonists seem to be rather useful at the neuromuscular junction in helping patients. And this child improved, and here he is after some time. And you can see his face is much more lively. He's able to smile much better, and apparently doing very much better. There was a poster yesterday which summarized some more cases, and in fact, we're keen to put together a whole international series of these patients. So if anybody knows of any others, I'm sure we'd like to hear about it. And you'll also hear more about the adrenergic agonists in neuromuscular transmission disorders from David Beeson and Jackie Pallis, and others this afternoon or later on today. So lastly, do we need to have those drugs then? We should be preventing the antibodies crossing the placenta and getting into the getting into the babies. That would be the best treatment once you know the mother's got an antibody that could be effective or previous pregnancies have been affected. One wants to stop those antibodies going across. And this is what this shows. The FcRn inhibitor that we used here was an anti-mouse FcRn inhibitor. As you know, those inhibitors reduce antibody levels, but they also block the transplacental transport of antibodies, of IgG, and that's shown here in an AChR antibody model with injected dams. And here are the embryo sera, and you can see that the inhibitors greatly reduce the amount of antibody that has crossed into the fetuses. So there is the potential there of treatments. Okay, this is my last slide. So I think myasthenia and the neuromuscular junction are still interesting. We don't know how retrograde signaling works. We don't know how salbutamol and ephedrine work. Can treatments be better targeted to the specific plasma cells instead of always being a general immunosuppression? Can SHP2 inhibitors be used in MuSK-MG? Pain in neuromyotonia should be better appreciated and asked about, in my view. And maternal antibodies can have long-term effects on the offspring, which could be perhaps avoided by preventing the placental transfer of those antibodies in the first place. And that is my last slide with my colleagues. And thank you very much for your attention.
Thank you very much, Dr. Visser. We have just time for one or a couple of questions.
Well, yeah, I'm sorry. I apologize for going on too long.
So over here. So a very nice talk. Is there anything known about regulation of SHP2?
Well, I certainly don't know anything about the regulation of SHP2. I think it's an important question. And what we need to do is we have already started, but to do more animal experiments to see what's actually happening to SHP2 and how much you can inhibit it in vivo. Thank you.
Another question? So thank you very much.
Thank you very much. And please, Professor Vincent, please stay on stage. Please stay.
Angela, thank you very much for all these contributions to this Congress. And as a present, can I have the slide, please? We had a very nice meeting in 1991.
Yes.
And John Newsom-Davies has been mentioned many times, and there were prominent speakers as Hohlfeld, John Newsom-Davies, Osterholz, and I'm the lucky possession of that script of that time, and I copied it for you.
Thank you.
And I want to give this to you.
Thanks very much.
So thank you very much for your attention, and we are closing this very interesting session. Goodbye. Thank you.